Thanks everybody for joining me. Good morning, good afternoon, good evening, wherever you are. We'll just jump right into things. So starting everything here with a very brief introduction. My name is Clayton. I have a background in genetics, and then I transitioned into bioinformatics for my thesis work. So my thesis work really focused more on structural homology modeling of proteins and viruses. So that's really when I entered the bioinformatics sphere. And you can see from the photo there that my hair hasn't always been as long as it is now. Like, as soon as this pandemic is over, my barber is going to need to take shears to me. But you guys aren't here to learn about me. You guys are here to learn a little bit about antibodies. So today I want to talk a little bit about an issue that I'm seeing a lot in the antibody industry, and that is specifically that antibodies are characterized based off of what they bind and not their actual physical properties. Now, this is a really, really big problem, and essentially is a big problem where antibodies haven't really been updated into the 21st century. The same standard wouldn't hold true. We wouldn't hold plasmids accountable, essentially, if we didn't have any sequence information for plasmids, for primers, for just general model organisms, then you can't publish absolutely anywhere. But for some reason, antibodies have really just kind of skirted underneath everything here and have a much more lax characterization. And that results in really a significant number of problems. So they're fairly well noted, and I'm just going to give a few examples here, starting with an example with a CDK1 antibody. So we've got here on the right a number of antibodies, all available from commercial sources, Sigma, Abchem, BD Biosciences. They're all advertised as reacting to CDK1. And they do a great job. They are very specific antibodies. They have a strong signal. So that's exactly what we want. But if you look over on the left, you have antibodies that are also being sold by Santa Cruz and also Abcam here that not only bind to CDK1, but they actually also bind to CEP152. And this is a really, really big problem here because both of these targets here are actually cancer biomarkers. And when we're not being able to realistically design any diagnostics, if it's not a monospecific antibody, and there's no way of distinguishing these because they're all just being advertised the exact same way as anti-CDK1 antibodies. And you can't even necessarily always trust just a single vendor to do everything. In both camps, we've got Abcam. So there's no good and reliable way to actually determine, is this going to be a good antibody or a bad antibody here? And this isn't kind of a cherry-picked example here. These specific antibodies have been referenced in over 200 papers. And we see the same problem across all kinds of different targets. So what we have got pictured here is an anti-ubiquitin antibodies, or several anti-ubiquitin antibodies from the heart and lung tissues. That's what the H and L are here. And you can see that they all show really, really different patterns here. So they're very clearly quite different, but you as a researcher have no way of actually being able to distinguish between any one of these antibodies. So how on earth do you go about trying to go and select any antibodies? And the fact of the matter is that researchers are wasting a ton of money and a ton of resources here working with all of these really bad antibodies. And there's no real way to actually distinguish between what a good antibody is and a bad antibody is because they're not characterized at all by their physical properties. They're just characterized based off of what they bind. And you can see in a lot of the actual sheets provided by the vendors themselves that the results aren't, they often reuse the same results over and over again. And instead of updating with every single batch, you essentially do the test once, and then they assume that that antibody is going to work the same forever. So that means that there are a ton of bad antibodies out there. And globally, people are wasting about $800 million on bad antibodies. And about half of that is all happening in the United States. So this is a really clear and pressing issue that's been going on absolutely everywhere. And these numbers are back from 2015. So the problem has only gotten worse. And we're investing significant amount of resources into antibodies that are bad, which are producing results that are non-reproducible. And it's a really difficult thing for the researchers to have to deal with here because through no fault of their actual own, they're just putting blind faith here into the antibodies that commercial vendors are supplying. And there are some very big significant problems here. And so how do you kind of go about trying to find good antibodies? I'm borrowing a slide here from a webinar hosted by Dr. Bradbury earlier this year. So as any good scientist would say, you go to the literature first. And when you're looking in the literature, you can see here we're looking at a CDK2 antibody, two of them. And you have the one on the left that has been used in about 690 publications as of a bit earlier this year. And the one on the right has been only used in a single publication. And on the face of it, when you look at some of this data, it looks almost as if they're pretty much the same. So you might as well go with the one that's been published a lot more frequently. But if you take a look at the scales, there's actually a tenfold difference between the two sequences, or between the two antibodies here. So when we adjust for that, you can see that the antibody that's only been sourced a single time is actually a far better antibody. But because they're only being advertised as this is a CDK2 antibody, there's no way of distinguishing between them. So researchers are typically just choosing the first antibody to be published, and they use that over and over again. So researchers are producing irreproducible results just because there's no good way of characterizing these antibodies. There's just so much unknown there that the researchers can't control whatsoever, and the industry really hasn't stepped up to address this issue. So the kind of typical thing that you would think to go about and actually solve this is, well, why don't we sequence the antibodies? And hybridoma sequencing comes to mind originally because most of these monoclonals are going to be made in hybridomas. And that's a really, really great starting point, actually. It's defining the antibodies off of their sequence is going to give us a much more robust, reliable, and repeatable results. But there's a kind of built-in problem here, in that about 30% of antibodies are actually found to have additional chains. So a study that took part over about 2 decades here and was published back in 2018 found with multiple different hybridoma sequencing companies that they were observing about 31.9% of antibodies have an additional heavy or light chain. And when we're talking about an additional light chain, we've got 3 possible variants here. We've got the so-called correct IgG, that's what you want, and that's going to be your actual good binder. And then you have one that's kind of a chimera between your additional light chain and the light chain you actually want. And that will still likely bind, just with lower affinity. But then when you can look at the kind of extreme where you have both of the non-desirable light chain here. And at best, that's going to bind nothing. And at worst, that's going to bind a completely different target here. And when you look at an additional heavy and light chain, then there's 10 possible variants in here. And only one of them is going to be the actual antibody that you want. And when you're doing hybridoma sequencing, there's actually no way of capturing this whatsoever. And these kind of mutations happen all of the time. You can have additional light chains from the essentially just some fusion with some multiple B cells. It might be that the original hybridoma mixture was non-clonal. The cell selected could be aneuploid, and kind of downstream, just in terms of maintaining your hybridoma here. You're going to have mutations throughout the entire process as you subclone that mixture. So there's going to be a huge amount of variability in here that just sequencing directly from the hybridoma isn't necessarily always going to address, and isn't going to capture any additional light chains at all. So how then do we even tackle this problem in terms of trying to figure out a good way to characterize the actual antibodies? Well, what if we could work off of the protein directly? It's if you have a tube of something that you know is already working, then if you could just have exactly that forever, you would be happy. But how do you sequence that? And that's exactly what Rapid Novor actually specializes in. So Rapid Novor is the world leading protein sequencing company in mass spec. So what we're doing is, instead of working from the DNA blueprint that isn't necessarily going to capture all of the information, we work directly off of the amino acids themselves. So that allows us to capture things like multiple light chains, because those light chains are going to be in the tube itself. And it also allows us to capture some kind of other notable observations, things like post-translational modifications, particularly additional glycosylation sites that are going to have really fundamental impacts onto the actual antibody itself. And that's really our specialization here. And how do we go about it? Well, we follow a fairly standard proteomics workflow here, at least on the face of it. So we take that sample that's been sitting in the back of your fridge for years and years and years, and you're very careful not to use too much because you're never going to get any more of it. But what we can do is essentially make sure that you have a guaranteed access to it forever. So you send us that sample, we're going to digest it with anywhere between about 5 and 10 different proteases that'll give us all kinds of peptides of varying lengths. And then those peptides are going to then be sequenced on the mass spec directly. That's going to give us the resulting peptides, which we can then go into our proprietary de novo assembly algorithm here to assemble a full sequence from N terminus to C terminus. And then we develop a full report for you. So that report's going to include the full sequence in FASTA format, as well as any kind of notable, bizarre observations that we've seen along the way. So even if on the face of it, that is a really standard proteomics approach, we do actually have a lot that makes us unique. We are hyper specialized here. So we exclusively actually do protein sequencing. And because of that, we are really, really good at it. So whereas a lot of teams don't necessarily struggle to get 100% coverage over an antibody, you can usually get that with actually a single different protease. So why do we bother using anywhere between 5 and 10? Well, ultimately, it's because our primary focus here is going to be accuracy. That's what sets us apart, kind of in a word here. We are using a very, very specialized cocktail of anywhere between 5 and 10 different proteases that are going to give us peptides of very, very different lengths and allow us to really optimize for everything here. So when we're looking at every single amino acid position, we actually have a minimum of 30 times coverage, and we're regularly seeing hundreds of peptides backing up every single amino acid position. And we want to make sure that there's evidence from multiple different proteases so that we can be completely confident in the actual results that we're delivering, that it's not just from a single peptide here that we're making a call that this might be a lysine. Essentially, we want to make sure that there is evidence from multiple different peptides derived from multiple different proteases. And then we can do all of this accuracy without even sacrificing any time. We can get this done really quickly because the bulk of this kind of initial analysis work is done using our proprietary machine learning algorithms. So these were developed by a professor from the University of Waterloo, Dr. Bin Ma, and who is also one of our founders and chief scientific officer. So using our proprietary machine learning techniques, we can assemble a draft sequence that can essentially get the N terminus to C terminus done with extreme levels of accuracy really, really quite quickly. And then one of our team of bioinformaticians here is going to go in and review every single amino acid position. And they're actually going to review the individual spectra themselves to make sure that we are completely confident in the sequence that we deliver to you. And we compound this with our proprietary WILD technology. So WILD stands for W-ion isoleucine leucine determination. And this is a problem in mass spec because when you're relying on a piece of technology that focuses on measuring the mass over charge ratio, and you have 2 amino acids that have the exact same molecular mass, that presents a really, really big problem here because you're breaking everything up along the peptide backbone, and you can't distinguish between the 2 of those in MS1. So what you can do is essentially fire it down the mass spec again, and the antibody or the amino acids are not going to break up exclusively along the actual peptide backbones, but they're actually going to break up the constituent R groups as well. And then those are going to lose a specific mass, which is coincidentally the W ion here. So from that, we can actually make an educated decision based off of data, whether we're observing a leucine or an isoleucine. And the reason this is really unique is because most teams really have to rely on looking at the germline sequence here. So what they'll do is they'll look in a database and see that, okay, about 30% of the time we're seeing an isoleucine here, and we're only seeing a leucine here maybe 2% of the time. So it's probably an isoleucine. But that's really just a best guess, and there's absolutely no data that's going into actually making that decision. So using this, we have concrete data to suggest that this is an isoleucine or this is a leucine. And that's really significant when you're looking at the CDRs because you're on average looking at between 4 and 5 different isoleucines, leucines in the CDRs. And if you're just guessing on those, then you might be expressing 25 potential variants here just to get one antibody that actually works. So using our proprietary WILD technique combined with our our experience with our experienced team of bioinformaticians and incredible depth of coverage here, we deliver a single sequence to you that is 100% accurate, so that you only need to express a single variant here. What we're trying to do is provide you with the best antibodies here, where hybridoma sequencing has challenges handling multiple different chains here, and the industry as a whole has a difficulty in terms of finding good antibodies. We want to make sure that once you have found a good antibody that you can continue to work with, you can base your research around that, and you only need to express one variant. And then once you have that sequence that's confirmed, that's been expressed, it is active, it is binding, then you already have a sequence there. So that immortalizes it for your research forever. And that means that you have reproducible results. The results can be reproduced by anybody absolutely anywhere, because you have that sequence information absolutely forever. And accuracy is incredibly important here, because if you're guessing at any individual amino acid position, then it's very easy to tell if you got the sequence wrong with an antibody, because it just won't bind. So accuracy is our main focus here, and we want to make sure that researchers aren't focusing on just assessing different antibodies or expressing all kinds of different variants here. We want to make sure that they can focus their efforts on their research itself and let us handle the actual antibody sequencing work here. And we've done that a lot. So we've worked with companies across the globe, over 300 organizations worldwide. We work with academics, we work with small businesses, medium businesses, and even some of the largest pharmaceutical companies here. And we've successfully completed over 1,600 protein sequencing projects for them. And well, what do they have to say about it? It's ultimately, we can give you the sequence, but that means nothing if it doesn't work. So just recently I was chatting with a client, and I was saying that we want to make sure that these antibodies are actually working. So what's your experience been with us in the past projects? And they found that the majority of antibodies were actually binding with equal or even better affinity here. And in some cases, you did have some antibodies that bound with slightly lower affinity. And this client was developing everything for kits here. But because they performed a little bit worse, but they had this immortalized sequence, it didn't really matter to them. They could just essentially modify the concentrations here and optimize the conditions so that it was still ultimately able to be converted into a commercializable kit. And they did actually ultimately have one project that they deemed a fail, where they couldn't actually produce an antibody that bound in really any meaningful amount. But because they had integrated us so early into their workflow and into their discovery process, they were able to essentially find out that this antibody was never going to be scalable, that they were never going to be able to actually bring this into a developable commercial product. So instead of investing all kinds of time, money, and resources into this project, they were able to abandon it very early on and just find a different target that actually was going to work and wasn't going to give the researchers such a headache throughout the entire experience. And this isn't unique. When we asked our clients, if you were talking to somebody that was considering working with RapidNovoR, what would you tell them? And they said that they would tell them how many I've sent to RapidNovoR and how many they've gotten right. And that's currently 7 out of 7. So that's a pretty good record. So ultimately here, what do you actually need to get started with a project for us? And the first thing that you're going to need is about 50 to 100 micrograms of purified antibody. And that's really it. That's all we need to actually start discussing a project here. We try and make it really, really easy to work with us, because our priority here is going to be to speed up research and discovery here. We want to handle the sequencing work so that you can immortalize your sequences, figure out exactly what is in that tube, express a single variant there, and have access to that antibody forever, so that you can focus all of your efforts onto your research, and we can handle all of the sequencing work here. So thank you so much for listening. Sorry, we got started a few minutes late, so we've only got about 5 minutes here for questioning. But if there's any questions here, then I'd be happy to answer them at this point. And if I don't get to all of them, feel free to send me an email. My contact information is just on the slide now.
All right, Clayton, so it looks like we have our first question, and it's, is it possible to characterize polyclonal antibodies?
That is an excellent question here. And the answer until very, very recently for us would have been maybe, but we actually really just recently actually developed a commercially available polyclonal sequencing service. So this is a fairly involved project, because when you're talking about a polyclonal antibody, it's a very complicated mixture of antibodies, and nobody knows necessarily how complicated it is. So it is certainly a much more involved project for us. But yes, we can actually handle polyclonal antibodies at this point, and we're really, really excited to have just unveiled that technology. It's very excited, and we just were able to actually complete our first project where we were able to actually verify the activity of a polyclonal antibody derived from rabbits. So there are a number of different challenges in that there's a number of different antibodies. You're trying to put a puzzle together that all the pieces are from different puzzles, but we've ultimately been able to address that very recently, yeah.
Okay, great. Another question is, can we handle samples that contain acidities?
Yes, ultimately what I'm talking about here in terms of a purified antibody is we need something that's been relatively purified. So as long as we've got about 80% your actual antibody of interest, then we can always work with that project here. So as long as it's primarily the actual antibody you want to have sequenced, then we'll run our protocol, and our algorithm is really, really sophisticated. And because of that, we can extract the peptides that are actually related to the antibody, and then ignore the peptides that are completely unrelated. So that means that we can handle samples with reasonably low purity, but as long as it's primarily the actual antibody you need, then we can handle it.
Okay, great. And then another one is, can we sequence proteins?
Yes, so it's always going to be dependent on the size here. So an antibody is a protein, so yeah, we can absolutely handle it. Antibodies have that really nice defined structure where you have the Fc and you have the variable domain. So it's our system is optimized towards sequencing antibodies, but we still do regularly sequence proteins more generally. Ultimately, it's going to be a more customized project here. So I'd recommend if you've got anything in mind, send me an email and we can discuss it. So we do the exact same protocol and we have the exact same de novo approach. And ultimately, it's just going to be based off of the size of the protein, and we should be able to handle it. But I'd be happy to discuss anything kind of in more specific details here over email.
Okay, and a couple more. Should the antibody sample be pre-decosylated, as glycosylated proteins are problematic to analyze with mass spec?
No. So you guys don't necessarily need to do any processing on your end. So we do treat our antibodies with a number of chemical modifications, which is we've spent many, many years developing everything over here. So you don't need to treat any, you essentially don't need to remove any glycans here. We will do everything. So in some cases, we do intact mass where we will remove the glycans, but you don't need to do any treatment here. We handle everything, and we have a number of techniques to handle difficult stuff on the mass spec. Sticky peptides, highly hydrophobic peptides. We spent a lot of time developing our protease digestions and our protocols here to be able to essentially get peptides and be able to accurately read them so that they can be fed into the automated assembly algorithm and reviewed for accuracy.
Okay, great. And probably the last question we'll be able to take today. Can you comment on Rapid's ability to delineate glycosylation sites across the whole antibody sequence?
So that's a really interesting question. So typically you're going to see some very specific glycosylation sites. So kind of position on the heavy chain, position 271, kind of plus or minus about 5 amino acids there. So typically we can see that, and we can actually identify the very specific residue that it is found on. So we have no issues with that. And fundamentally, using our algorithms here, we do actually pinpoint the individual amino acid that the glycan is attached to. So we do have everything optimized here. I can't go too much into details here in terms of the technology that goes behind it, but we do delineate the glycosylation sites. So if we're seeing different N or O-link glycans, we'll not only tell you that they are present, we'll tell you the specific residue to which they are bound.
All right, and I think that wraps up the questions that we can take today. So.
Okay, perfect. Well, thank you everybody for coming here today. If you have any other questions, feel free to send me an email at any point, and I'd be happy to answer them. And I'll let you guys all have a great weekend. Thank you so much for coming. Have a great day.